So you can hear, right? Everything okay?
Okay.
Okay, yeah, I start. So good morning, everybody. It's nice to have this online meeting for this very special period. I really appreciate for the organizer to make the effort to organize this event. I think it's very interesting, very meaningful. Today I would like to present this topic: the molecular properties of fungal polysaccharide and their bioactive functions associated with the gut microbiota. So I'm from the Hong Kong Polytechnic University, the Department of Applied Biology and Chemical Technology. So today I want to cover these contents. The first, I will give a brief review on the gut microbiota in human health and disease, and then on the properties and bioactive function polysaccharides from medicinal fungi. And the last part, the main part of my presentation, is about the polysaccharide, extracellular polysaccharide from mycelial fermentation or medicinal fungus, CSHK1, on their molecular properties and the prebiotic functions. So first of all, gut microbiota, I think is probably you already heard, about gut microbiota. Human gut microbiota is referred to the entire microbial community colonizing human gut, which is mainly composed of about trillions, more than trillion of bacterial cells in the large intestine. So you can see this diagram. You can see the number of bacteria change in the large colon, large intestine. So from the the proximal colon is about in this range 10 to the 10 to 11, then gradually increase 10 to 11 to 10 to the 12. Then the distal colon, the largest number over 10 to the 12. And so also the change in the pH condition inside the large intestine or in the colon. So of course this number just rough estimate, just saying, so actually there is a large, a huge number of bacteria in our gut. But this bacterial population act as the microbial organ, making many important contribution to the host physiology and nutrition, including metabolism and the protection. They have these kind of functions. First, the microbiota is responsible for the fermentation of dietary fibers. And also the fermentation of dietary fiber can generate energy for the local cells. And then also can synthesize vitamins, which are very important nutrients for the host, and also responsible for the digestion of harmful compounds, or this called detoxification function. Then also have the protect effect, including immune stimulation, and also produce antimicrobials, and have control against the bacterial pathogens, to prevent the proliferation of the pathogenic bacteria, and so on. So actually, the gut microbiota, they also had a close connection with many human diseases. Is a dysbiotic or abnormal gut microbiota is implicated in several important human diseases, such as inflammation and metabolic disorders. So this is the a diagram to show the in the dysbiotic microbiota is associated with decay, epithelial. So this is the barrier function will be weakened. So then the leading to the leakage, the entrance, of the bacterial components, and whole bacteria into the bloodstream, then cause a lot of disease and the disorders, including inflammation, in local gut inflammation, also in other part of the body, even to the in the brain. So anyway, this is about the polysaccharide. Polysaccharide in the living organism. I think we all know about polysaccharide. Okay, they are represent a major class of biomicromolecules. In addition to other macromolecules in the living organisms, such as proteins and nucleic acid. So, but polysaccharides are more diverse and more complicated, more variable in their structure and properties. So the common biological function, the most common, there are two major functions. One, they are structural component of the cell walls. Okay, such as this. Okay, this is a fungal cell. Can see that. Okay, beta glucan and the chitin. Okay, they form the major component of the cell wall structure. And then they also have another function, nutrient function. Okay, like starch. Okay, like starch and the glycogen. Okay, so they are the major nutrients and energy source. Okay, so then of course they are also structural component in the plant. Okay, even in the animal. Okay, so they also present. So then the structure features. Okay, polysaccharide, the biomicromolecule, they have several order of structure, primary, secondary structures. Okay, and also have high order structure, tertiary, and high order structure such as the conformation and the random coil, triplet helix conformation. Also in water, okay, mainly in water, okay, they form aggregate and gel. Okay, this also formation of the gel and aggregate will significantly affect the property of the solution, such as viscosity and also other functional properties of the solution, such as the texture of food product. And in addition to this structure, other molecular property characteristics, okay, may also affect their function and bioactivity, such as molecular weight. Okay, molecular weight. So as polysaccharide, they have very high molecular weight, but the previous study have shown that relatively high molecular weight are required for their bioactive function, such as immuno bioactive function. And another one is the degree of branching. Okay, so most of bioactive polysaccharides have side chains, such as beta glucans. Okay, they have usually the active beta glucan, they have branches. So then polysaccharide, the metabolite, okay, they have because many of the bioactive polysaccharides, they are non-digestible. They will pass through the digestive tract. They will get into the large intestine. Okay, then they will can be metabolized. Okay, fermented by the microorganism, bacteria. Okay, so but this, okay, they can be consumed as the nutrients, such as the diet fiber, and or another term called prebiotics. Okay, so consumption of this diet fiber, prebiotics. Okay, these are mainly polysaccharide. Okay, or oligosaccharide. It's an important dietary strategy for modulating microbiota. So it's actually sometimes our microbiota, it's out of order. Okay, we can take the diet fiber and prebiotics, maybe can recover microbiota to normal state. And also the selective utilization of various common polysaccharide by different microbial species in the gut microbiota. So because there are many different species of bacteria in the microbiota, they can selectively consume, utilize different polysaccharide, which will result in the change in the microbiota composition. Another aspect is the fermentation non-digestible polysaccharide can produce the short-chain fatty acid SCFA, so which serve as the nutrients for the gut epithelial cell and also play many other important local and systemic functions. This slide shows more specifically the physiological and the metabolic functions of the short-chain fatty acid. Okay, the first one 13 fatty acid, the major metabolic product from the fermentation of the polysaccharide and diet fiber. Okay, they can act as the energy source for local and distant tissues, and they are also the major signal molecules for the host immune system, and also for the development and function. Okay, another one is regulating the function to regulate energy homeostasis and metabolism in the human body. Then, of course, this show okay the diagram to show okay the metabolism of the diet fiber and the prebiotics. Okay, by the microbiota generate short-chain fatty acid. Okay, these are three major short-chain fatty acid, so they can get into the bloodstream. Okay, then can interact with the receptors. Okay, including G protein coupled receptor and the G protein coupled receptors, then result into various responses such as blood pressure, regulating the blood pressure, immunity, and the cell cycle, and also other physiological and process inside the human body. So then further about the polysaccharide from the edible medicinal fungi. So polysaccharide represent the major cloud bioactive molecule in edible medicinal fungi or mushroom. So in most the mushroom, okay, the polysaccharide had the content around about 5 to 10%. That's the common value. Okay, by weight is represent the most abundant micromolecule in the mushroom. So they have notable anti-tumor and immunomodulatory activity. They also have many other activities such as hypoglycemic, hypolipidemic, antioxidant, anti-inflammation, antiviral, and so on. So of course, this activity depends on their structures. So the common bioactive fungal polysaccharide structure that are most well known include the beta glucan and beta polysaccharide protein complex. So this diagram shows some of actually many of the functional food or dietary supplement on the market, and also therapeutic agents composed of polysaccharide from medicinal fungi, edible medicinal fungi. Okay, so you can see this already. Okay, the product on the market. So mushroom. Okay, recently the many scientists and the researchers have indicate that mushroom can be mushroom polysaccharide can be potential prebiotics. What are prebiotics? Prebiotics are the non-digestible food components, very similar, just diet fiber. So but this diet fiber, they can be fermented. They can be utilized by the gut bacteria. So they can enter the large intestine and selectively enhance the growth activity of certain bacteria within the gut, within the bowel, which will lead to a beneficial effect to the whole health. Mushroom, they are promising source of probiotic with abundant bioactive polysaccharide, including chitin, beta, and other glucans, mannite, xylane, and so on. There are many non-digestible polysaccharide. Also, they are bioactive. Their bioactivity may be related to the gut microbiota. So actually, this is the not recent already. The 10 years ago, a review paper. It's saying that mushroom has the potential source of prebiotics. So now this is the meeting summary. So this is the meeting summary. The summary conclude that. So the beneficial effect of functions of mushroom are associated with the gut microbiota. And also the immune functions are related to the microbiota. Of course, the immune function of mushroom are mainly attributed to the polysaccharide. sorry about the interruption. Okay, this is the recent review paper. Okay, so also indicate that mushroom, the health benefit of mushroom, okay, are associated with the gut microbiota. You can see the mushroom polysaccharide, they have this various health benefit, including anti-hyperglycemia and lower oxidative stress, and in the animal, and also other reduced obesity and anti-diabetic and so on. Okay, we're also the anti-inflammation and the therapeutic effect on the IBD. Okay, that's the intestinal the bowel, okay, the inflammatory bowel disease. Okay, so this activity, these benefits are probably associated with their effect on the microbiota. Okay, so actually studies already shown that this polysaccharide can alter the gut microbiota microflora composition, also change the composition and activity of the intestinal microbiota. This activity, this effect are probably associated with their health benefit of the polysaccharide. So still, actually, this is a very complex relationship. Still, we do not really know very clearly how this health benefit are linked to their effect on the microbiota. There's still a lot of uncertain answer question need further research and study. So then come to my the last part of the talk is on this very interesting medicinal fungus. The name called Cordyceps sinensis. Okay, so the general name called Chinese caterpillar fungus, or 冬虫夏草 in Chinese. The meaning of 冬虫夏草 means winter worm, summer grass. Okay, so the natural species look like this. Okay, actually they're the insect, the larva. Okay, on the top that's the fruit body. Okay, the fungal fruit body of the infection of the whole insect, then form this fruit body, the whole complex called winter worm, some grass. Okay, then naturally they are mainly found in the high mountain Qinghai Tibet plateaus. Okay, in that area, very high level. Okay, in Western China, probably also found in the near region like India and in that region. Okay, so taxonomically, this the fungus is called Cordyceps sinensis. Okay, taxonomic name called sinensis. That's the old name. The new name called Cordyceps sinensis. Okay, it's the insect parasitic fungus. So this diagram shows the formation process of this amazing, very interesting fungus, insect fungus complex. Okay, it shows the cycle. Okay, so usually the larva, the insect, will be buried, underground, stay underground. At that time, the fungus with the spore will infect, will fall on the insect. Okay, then start the infection. Okay, then through this from October and November until next May or June. Okay, so then form this complex. Okay, take about 8 to 10 months. Okay, then of course the local people can find out, can dig out this this organism. Okay, it had a very high price. So of course, this medicinal fungus have been used in traditional medicine, also traditional Chinese and Tibetan medicine for centuries. Okay, so there have a lot of therapeutic and health benefits. And this one show our production process. Actually, this is the CHK1, the fungus isolated from the fruiting body of this natural Cordyceps sinensis. Okay, after we identify the fungus. Okay, we start to establish the mycelia culture. Okay, this show the appearance of this CHK1 mycelia on the solid medium, and then we usually in the lab we cultivate this in the liquid medium in shake flask. Of course, we also can use large scale use fermenter to grow the fungus. So at the end of the culture, it take about 6 or 7 days. At the end, we just do centrifugation to separate the mycelium biomass and the liquid medium. Then we use ethanol precipitation. You can see the cotton-like the stuff on the surface. This is the EPS extra cellular polysaccharide. Then we do further purification. This is the crude. The total sugar about 70%. Also contain a lot of protein. And after purification, we remove the protein and also remove the small molecules. We can get about higher content total sugar and the lower content of protein. So this polysaccharide mainly composed of 3 monosaccharide. The glucose is the most abundant monosaccharide in this extracellular polysaccharide EPS. So actually, in one of our studies, what we did in order to study the probiotic function of EPS, and another, we also use a plant, polysaccharide, and the correlation to their molecular property, especially the molecular weight. This is the way show that. So after we isolate the polysaccharide, so use the ethanol precipitation, we use 2 ways to treat the polysaccharide. The polysaccharide has a high molecular weight. We use ultrasound, power ultrasound or use acid degradation to lower the molecular weight of polysaccharide so that we can generate lower molecular weight fractions of these 2 EPS and KGM. KGM is a very well-known plant polysaccharide contact glucomannan. It's also a well-known diet fiber from a contact plant tuber. So we actually study just we did a study to compare our polysaccharide with the KGM in their probiotic functions. So actually this showed our result, the measurement of the molecular weight use the gel permeation chromatography. This showed the profiles actually. So actually this show the retention time. The peaks are the molecular weight. Actually the EPS have several peaks. That means composed of the different molecular weight fractions. So elution time, for the GPC, the higher molecular weight, come out earlier. The lower retention time means higher molecular weight. Longer retention time means lower molecular weight. This show the result, the molecular weight measurement, the EPS, you can see the very high molecular weight, before the treatment. And after the ultrasonic treatment, the molecular weight reduced to about 10 to the 7th, but still ultrasound is limited, it had a limited effect, so then we use acid, of course, you can use acid to degrade the polysaccharide, more extensively, depending on time and temperature, we could generate a very relatively low molecular weight polysaccharide fractions. So so you can see that with acid degradation we can get to a few thousand molecular weight. In general, for power ultrasound, is a very convenient way, the mechanical method to degrade the polysaccharide. So with treatment by power ultrasound, it can increase the water solubility, can also reduce the molecular weight, moderately, can also reduce the solution viscosity, because viscosity is correlated to the molecular weight. Sorry. So then we test this polysaccharide and also KGM in the pure culture. Actually, with several species of bifidobacteria. Bifidobacteria represent a major group of beneficial probiotic bacteria. So this one we did an individual culture of the bifidobacteria to see how the EPS affect the growth and the viability of the bifidobacteria. Okay, so actually our results show that the EPS, even after ultrasonic degradation, okay, was not really metabolized by the bacteria, okay, because only one single bacteria probably do not have all the enzyme to metabolize the complex polysaccharide, okay, even with the KGM and also the acid degraded polysaccharide or KGM, okay, so the bacteria cannot use the polysaccharide as a carbon source for growth. But a very interesting effect we found. So actually the polysaccharide can protect the bacteria, can sustain the viability of the bifidobacteria. You can see that this water we measured, okay, so the colony formation units of the bifidobacteria, you can see that this is the bacteria, okay, grown in glucose medium, glucose and the major carbon source. These are the two grown in, okay, in the EPS, okay, medium. This is another one, EPS with ultrasonic treatment. This is EPS without ultrasonic treatment. So what we did that we grew the bifidobacteria in the medium for 24 or 48 hours. Then we inoculate the bacteria on the solid culture. This is what we observed. Okay, after 24 hours, you can see that the bacteria previously grown in the glucose media had a very few spores and a very low viability. But with EPS, the viability was significantly higher. Okay, so that this showed that the EPS had a strong protective effect on the bifidobacteria in the culture. Okay, also KGM also had protective effect, but the EPS is much stronger. So then this one showed how EPS affect the bifidobacteria, or also the probiotic bacteria, also include the lactobacillus bacteria. Okay, during cold storage at 4 degrees Celsius. Okay, for a long period of time. Okay, this one in the simulated gastric acid to simulate the gastrointestinal conditions at the low pH. Okay, how they affect that. Okay, affect the bacteria viability. So you can see that the blue one show the bacterial culture supplied with EPS. Okay, so we also have other like the GOS, the oligosaccharide. Obviously, with the EPS, you can see the blue one. Okay, for most of the bacteria, the viability can sustain for much longer time. Okay, so that our results show that the EPS can protect the bacterial cell, the bifidobacteria and the lactic acid bacteria under stressful conditions. So this one we estimate the reduction of the death rate during cold storage and gastric acid treatment. You can see that with the EPS, the death rate can be reduced by 50 to 70%. Okay, so this significant effect. Other than inulin is also regarded the well-known probiotic. GOS also probiotic, so their effects are much weaker or smaller. So then another one which we test the effect of bacteria. Okay, the bacteria were treated by antibiotics. You know the antibiotics are inhibitory, toxic, or can inhibit the bacterial growth. Okay, can kill the bacteria. Okay, so we use several the common antibiotics include penicillin, tetracycline, and streptomycin. Okay, so we measure the minimum inhibition concentration and also minimum bacterial concentration. So you can see with the supplementation of 5 gram EPS, all the value MIC, MBC are all much higher. Okay, so at this concentration much higher than the control and also higher than in the inulin. Okay, so also this protective effect change with the concentration okay, so the concentration increase from 1 gram to 5 gram, the protective effect increase. Okay, so our finding is that, okay, the EPS can protect the bifidobacteria against the antibiotics. So then the previous results were obtained in pure culture of individual, okay, probiotic bacteria. Then we also did, okay, the human fecal fermentation. So this okay, the human fecal is more, okay, is similar, okay, to the human gut microbiota. Okay, so this is what we did. We just we did the fermentation in the okay, small tubes. Okay, so with the human fecal samples. Okay, so at a given temperature, then we use the method to I mean to measure, okay, the growth and viability of different group of bacteria in the human fecal samples of the, okay, 24-hour fermentation under anaerobic condition. So that you can see that, okay, they show that the control without any, okay, EPS, without any additives. Okay, so they show the, okay, so after 24 hours, okay, the show the, okay, log 10, okay, line. We use this one to represent, okay, the population, okay, at this level. But after we add EPS, okay, the population, okay, the number were much higher for most of the bacteria species, right? So the major group bacteria, that means EPS can be nutrients for the fecal bacteria. Okay, so they see because they actually, our later study also show that EPS could be metabolized by the fecal bacteria. So you can see the, okay, the content, the number increase. Okay, so but if we apply the antibiotic, can inhibit the bacteria, fecal bacteria. Okay, so at the very high concentration, then all the fecal bacteria were killed. Okay, so then, but with the supplementation of EPS, so you can see that, okay, can protect. Okay, so we can see still see, okay, relatively high, okay, number of the, okay, bacteria, fecal bacteria population. Okay, you can see that this result show that, okay, EPS could also protect, okay, the fecal bacteria, okay, in the complex, okay, fecal bacteria system. So another, okay, the study we studied, okay, the metabolism EPS. Okay, so this the results show that, okay, actually we use, okay, the different polysaccharide fraction. This EPS, the original, the native EPS, isolated from the fermentation broth. Okay, so the intrinsic viscosity, which correlates to the molecular weight. Okay, so then we use EPS U1, U2, U3. This is the EPS degraded by the power ultrasound for 0.5, 1, and 3 hours. You can see the intrinsic viscosity gradually decrease. Okay, intrinsic viscosity is proportional to the molecular weight. That shows the, okay, gradual reduction of average molecular weight of the polysaccharide. So our objective is to test, to evaluate the dependence of the effect of polysaccharide, the dependence on the molecular weight. Okay, so actually we did see that. You can see that, okay, so they see the with the EPS, the highest molecular weight, this with the lower molecular weight. Okay, so also for the, okay, this consumption, okay, so lower molecular weight consume much faster than the higher, and the lower molecular weight was consumed much faster than the higher molecular weight. Also produce higher concentration of the short-chain fatty acid. Okay, but just initially, okay, if we keep the culture for a long time, then they all reach the same level. Okay, so all the EPS could be utilized by the fecal bacteria. Okay, so that they show that the EPS can be fermented, okay, by the fecal bacteria, can produce short-chain fatty acid. Okay, short-chain fatty acid are beneficial to the, okay, the host health. Okay, as I have shown previously. So major findings, okay, this is the major lower molecular weight polysaccharide were more quickly consumed by fecal bacteria. So then another one, the finding, we also measure the population distribution of the fecal microbiota, okay, through the genomic method. Okay, so you can see that with different MC, the control is the methylcellulose, is the inert, okay, polysaccharide, which cannot be utilized. We use that the control, okay, so that after supplement of different EPS, okay, they have different effect, result in different population distribution, composition, or relative abundance of the bacteria and the fecal bacteria. Okay, so this show that our result just show the influence of EPS fraction on the population distribution or on the composition of the fecal microbiota was molecular weight dependent. So that's the end of my presentation, a brief summary. Okay, so my presentation has shown that polysaccharides from additive management fungi have valuable property, function, and bioactivities. Okay, so then the bioactivity and health benefit of polysaccharides may have a close mechanistic connection with the gut microbiota. The EPS from this CSH1 mycelium fermentation had a protective effect on probiotic bacteria, also on the fecal microflora. The EPS showed a significant and molecular weight dependent probiotic effect in the fecal fermentation. So at the end, finally, I would like to make an acknowledgement. We would like to thank the contributors, my students, my collaborators to this work, and also acknowledge the financial support from our university, government, and from also Shenzhen government. So, and also the last but not least, I would like to thank you for your kind attention. And I wish you all the best, the good health and a peaceful life. Thank you, that's all.
Thank you so much, Professor Wu from Hong Kong. So I'll leave some time here. If anyone have any comments or questions, you can go and please ask Professor Wu.
Good morning. Can you hear me?
Morning.
I have a question.
Okay.
The presentation was really interesting, and I just would like to know, how did you perform the in vitro test to test that there was an increase of a probiotic? So how did you perform? You put EPS inside the media here? Yes, exactly.
Oh, this one. Oh, yeah. Okay. Yeah. So actually, it was very simple. Actually, this is actually the culture tube we use. Okay, but of course, for the bifidobacteria, very clean. Okay, so in the individual cell, we just like we grow the other bacteria. We inoculate the bacteria in the liquid medium, and then we just. But everything must be under anaerobic conditions. So we usually put this in a jar. Okay. In an anaerobic jar, we also had to supply anaerobic sachet to maintain anaerobic atmosphere. Then we place the jar, place the tubes on a shaker. Then we shake, okay, during the culture period. For this test, we did on different bifidobacteria. So we culture the bacteria for 24 hours. Then we inoculate the bacterial cells on the solid culture to test the CFU. So this was so you can see that we test in different, with different carbon source. Okay, so this is what we did. Is that clear?
Thank you.
Yeah.
Yes, thank you so much.
Thanks. Actually, our culture procedure was quite simple. I think most of our study have been published. If you are interested, you can search for my publication. All the procedures have been described in detail.
Thank you.
Thanks.
Thank you, Professor. Thank you.